यह है फोटोसिंथेसिस इन द हायर प्लांट्स। इसमें इसमें देखते हैं कि all animals, including the human beings, depend on the plants for their food. Have you ever wondered from where plants get their food? Green plants, in fact, have to make or rather synthesize the food they need, and all other organisms depend on them for their needs. The green plant make or rather synthesize the food they need through the photosynthesis and are therefore called as the autotrophs. We have already learned that the autotrophic nutrition is found only in the plants, and all other organisms that depend on the green plant for food are heterotrophs. Green plants carry out photosynthesis in a physiochemical process by which they use the light energy to drive the synthesis of the organic compounds. Ultimately, all living forms on the earth depends on the sunlight for the energy. The use of the energy from the sunlight by doing photosynthesis is the basis of the life on the earth. Photosynthesis is important due to the two reasons. It is the primary source of the food on the earth. It is also responsible for the release of the oxygen into the atmosphere by the green plant. Have you ever thought what would happen if there were no oxygen to breathe? This chapter focuses on the structure of the photosynthetic machinery and the various reactions that transform the light energy into the chemical energy. फिर आता है व्हाट डू वी नो। तो कहता है कि लेट अस ट्राई टू फाइंड आउट व्हाट वी ऑलरेडी नो अबाउट द photosynthesis. Some simple experiment you may have done in the earlier classes have shown that chlorophyll, green pigments of the leaf, light and the CO2 are required for the photosynthesis to occur. You may have carried out the experiment to look for starch formation in the leaf. To leaf, a vegetated leaf or a leaf that was partially covered with the black pepper and exposed to the light. On testing the leaf for the presence of the starch, it was clear that the photosynthesis occurred only in the green parts of the leaves in the presence of the light. फिर कहता है कि another experiment you may have carried out where a part of the leaf is included in a test tube containing some QH soaked cotton which absorbs the CO2 by the other half leaf is exposed to the air. The setup is then placed in the light for some time. On testing for the presence of the starch later in the two parts of the leaf, you must have found that the exposed part of the leaf tested positive for the starch while the portion that was in the tube tested negative. This shows that CO2 was required for the photosynthesis. Can explain how this conclusion could be drawn. फिर कहता है early experiments. So it is interesting to learn about those simple experiments that led to the gradual development in our understanding of the photosynthesis. Joseph Priestley in the 2020 performed a series of the experiments that revealed the essential role of the air in the growth of the green plants. Priestley, you may recall, discovered oxygen in the 1774. Priestley observed that a candle burning in the closed space, a bell jar, soon gets extinguished. Similarly, a mouse would soon suffocate in a closed space. He concluded that a burning candle or an animal that breathed the air both somehow damaged the air, but when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley hypothesized as follows: plant restore to the air whatever breathing animals and the burning candles remove. फिर कहता है कैन यू इमेजिन हाउ प्रिस्टली वुड हैव कंडक्टेड द एक्सपेरिमेंट यूजिंग अ कैंडल एंड अ प्लांट. Remember, he would need to rekindle the candle to test whether it burns after a few days. How many different ways can you think of light to the candle without disturbing the setup? Using a similar setup as the one used by the Priestley, but by placing it once in the dark and once in the sunlight, Jane Ingenhousz showed that sunlight is essential to the plant process that somehow purifies the air followed by the burning candle or breathing animals. Ingenhousz, in an elegant experiment with the aquatic plant, showed that in the bright sunlight small bubbles were formed around the green parts while in the dark they did not. Later he identified the bubbles to be of the oxygen, hence he showed that it is only green parts of the plant that could release the ऑक्सीजन. तो ये है प्रिस्टली का एक्सपेरिमेंट. प्रिस्टली का एक्सपेरिमेंट इस तरह से दिखाया हुआ है. जब कैंडल रहा तो माउस डेड हो गया, लेकिन जब मिंट प्लांट को रख दिया तो माउस है जो अलाइव रहा. फिर आता है कि इट वाज नॉट until the 1854 that the Julius Bonsack provided an evidence for the production of the glucose when the plant grew. Glucose usually stored as the starch. He later studies show that the green substance in the plant, chlorophyll as we know it now, is located in the special body later called as the chloroplast within the plant cell. He found that the green parts in the plant is where glucose is made, and that the glucose usually stored as the starch. Now consider the interesting experiment done by the T.W. Engelmann. Using a prism, he split light into its spectral components and then illuminated with a green alga cladophora placed in the suspension of the aerobic bacteria. The bacteria were used to detect the fact of the O2 evolution. He observed that the bacteria accumulated mainly in the region of the blue and the red light on the split spectrum, a first action spectrum of the photosynthesis by thus described. It resembles roughly the absorption spectra of the chlorophyll A and the B. By the middle of the 19th century, the key features of the plant photosynthesis were known, namely that the plant could use light energy to make the carbohydrates from the CO2 and the water. The empirical equation representing the total process of the photosynthesis for the oxygen evolving organism, by then understood as CO2 plus H2O लाइट दिए तो CH2O ग्लूकोज प्लस O2 बन गया. फिर आता है कि when the their glucose is represented as a carbohydrate, glucose a fixed carbon sugar. फिर आता है a milestone contribution to the understanding of the photosynthesis by that made by the microbiologist Collius Van Neel, who based on his study of the purple and the green bacteria demonstrated that photosynthesis is usually a light dependent reaction in which hydrogen from a suitable oxidable compound reduces carbon dioxide to the carbohydrates. This can be expressed by H2A plus CO2 और लाइट के प्रेजेंस में 2A plus H2O plus H2O बन गया. फिर कहता है in the green plants H2O is the hydrogen donor and the oxide to the O2. Some organisms do not release O2 during the photosynthesis when H2S instead is the hydrogen donor for the purple and the green sulfur bacteria. The oxidation product is the sulfur or the sulfate depending on the organism and not the O2. Hence, we infer that O2 evolved by the green plants comes from the H2O, not from the carbon dioxide. This was later proved by using radioisotopic techniques. The correct equation that would represent the overall process of the photosynthesis is therefore कि CO2 plus 12H2O लाइट दिए तो 6 plus 6 CO2 plus 6O2 बन गया. तो ये करेक्ट इक्वेशन है. Where C6H12O6 represents the glucose, the O2 release is from the water. This was proved by using the radioisotopic technique. Note that this is not a single reaction but description of a multi-step process called a photosynthesis. फिर आता है where does the photosynthesis take place. तो you would of course answer in the green leaf or in the chloroplast based on the part you read earlier in the chapter 8. You are definitely right. Photosynthesis does take place in the green leaf of the plant, but it does so in the other parts of the leaf also. You would recollect from the previous unit that the mesophyll cells in the leaves have a large number of the chloroplasts. Usually the chloroplasts align themselves along the walls of the mesophyll cells such that they get the optimum quantity of the incident light. When do you think the chloroplasts will be aligned with their flat surface parallel to the walls? When would they perpendicular to the incident light? You have studied the structure of the chloroplast in the chapter 8. Within the chloroplast there is a membrane system consisting of the grana, the stromal lamella and the matrix. There is a clear division of the layer between the chloroplast. The membrane system is responsible for trapping the light energy and also the photosynthesis of the ATP and the NADPH. In the stroma enzymatic reaction synthesize sugar which in turn form the starch. The former state of the reaction, since they are directly light driven, are called as the light reaction, photochemical reaction. The latter are not directly light driven but are dependent on the products of the light reaction ATP and the NADPH. Hence, to distinguish the latter, they are called as by convention as the dark reaction, carbon reactions. However, this should not be construed to mean that they occur in the darkness or that they are not light dependent. तो ये दिया हुआ है डायग्रामेटिक रिप्रेजेंटेशन ऑफ द इलेक्ट्रॉन माइक्रोग्राफ ऑफ अ सेक्शन ऑफ द क्लोरोप्लास्ट दिया हुआ है. इसमें जो है आउटर लैमली है, आउटर मेम्ब्रेन हो गया, आउटर इनर मेम्ब्रेन हो गया, स्ट्रोमल लैमली हो गया, ग्रेना हो गया, स्ट्रोमा हो गया, राइबोसोम हो गया, ये हो गया स्टार्च ग्रैन्यूल और ये इधर हो गया लिपिड के ड्रॉपलेट्स दिखाए हुए हैं. फिर आता है हाउ मेनी टाइप्स ऑफ द पिगमेंट आर इन्वॉल्व इन द photosynthesis. So looking at the plant, you have ever wondered why and how there are so many shades of the green in the leaves even in the same plant? We can look for the answer to this question by trying to separate the leaf pigments of any green plant through the paper chromatography. A chromatographic separation of the leaf pigments show that the color that we see in the leaves not due to a single pigment but due to the four pigment chlorophyll A bright or the yellow. In the chromatogram, chlorophyll B yellow, green, xanthophylls yellow and the carotenoids yellow to the green orange. Let us see what various pigments play in the photosynthesis. Pigments are substances that have an ability to absorb the light at a specific wavelength. Can you guess which is the most abundant plant pigment in the world? Let us study the graph showing the ability of the chlorophyll A pigment to absorb the light of the different wavelength. Of course, you are familiar with the wavelength of the visual spectrum of the light as well as the behavior. From the figure 13.3a, can you determine the wavelength color of the light at with the chlorophyll A for the maximum absorption? Does it show another absorption peak at any other wavelength too? If yes, which one? फिर कहता है कि now look at the figure 13.3b showing the wavelength at which the maximum photosynthesis occurs in a plant. Can you see that the wavelength at which there is a maximum absorption of the chlorophyll A, that means in the blue and the red regions, also for higher rate of the photosynthesis? Hence, we can conclude that chlorophyll A, the chief pigment associated with the photosynthesis. But by looking at the figure, can you say that there is a complete one-to-one overlap between the absorption spectrum of the chlorophyll A and the action spectrum of the photosynthesis? तो ये दिया हुआ है एब्जॉर्प्शन स्पेक्ट्रम दिए हुए एब्जॉर्प्शन स्पेक्ट्रम एब्जॉर्प्शन ऑफ द लाइट बाय द क्लोरोप्लास्ट पिगमेंट। ये हो गया क्लोरोफिल बी का और ये हो गया कैरोटिनॉइड्स का और ये हो गया क्लोरोफिल ए का, ब्लू वाले क्लोरोफिल ए का है। फिर आता है रेट ऑफ द फोटोसिंथेसिस मेजर्ड बाय द O2 रिलीज। तो यहाँ पर भी देख रहे हैं कि ब्लू लाइट में और रेड लाइट में जो ज़्यादा है लेकिन जीरो नहीं हुआ है। फिर आता है लाइट एब्जॉर्बड का देखेंगे और रेट ऑफ द फोटोसिंथेसिस देखेंगे तो एब्जॉर्प्शन यहाँ पर जीरो हो गया लेकिन अभी भी एक्शन जीरो नहीं हुआ है। लाइट एब्जॉर्बन का देख रहे हैं, वेवलेंथ ऑफ द लाइट इन नैनोमीटर्स में देख रहे हैं। तो कहता है कि ग्राफ शोइंग द एब्जॉर्प्शन स्पेक्ट्रम ऑफ द क्लोरोफिल ए एंड द बी द कैरोटिनॉइड्स, ग्राफ शोइंग द एक्शन स्पेक्ट्रम ऑफ द फोटोसिंथेसिस, और फिर कहता है ग्राफ शोइंग द एक्शन स्पेक्ट्रम ऑफ द फोटोसिंथेसिस सुपर इम्पोर्ट ऑन द एब्जॉर्प्शन स्पेक्ट्रम ऑफ द क्लोरोफिल ए। फिर कहता है कि these graphs together show that most of the photosynthesis take place in the blue and the red regions of the spectrum. Some photosynthesis does not take place at the other wavelength of the visual spectrum. Let us see how this happens. Through the chlorophyll is the major pigment responsible for trapping light. Other thalochrome pigments जैसे chlorophyll B, xanthophyll and the carotenoids, which are called accessory pigment, also absorb the light and transfer energy to the chlorophyll A. Indeed, they not only enable a wider range of the wavelength of the incoming light to be utilized for the photosynthesis but also protect chlorophyll A from the photo oxidation. फिर आता है भाटी द लाइट रिएक्शन। तो लाइट रिएक्शन और द फोटोकेमिकल फेज इंक्लूड द लाइट एब्जॉर्प्शन, वाटर splitting, ऑक्सीजन रिलीज एंड द फॉर्मेशन ऑफ द हाई एनर्जी केमिकल इंटरमीडिएट्स। ATP and the NADPH several protein complexes are involved in the process. The pigments are organized into the two discrete photochemical light harvesting complexes LHC within the photosystem 1 and the photosystem 2. These are named in the sequence of their discovery and not in the sequence in which they function during the light reaction. The LHC are made up of the hundreds of the pigments molecule bound to the protein. Each photosystem has all the pigments except one molecule of the chlorophyll A, forming a light harvesting system also called as the antenna. These pigments help to make photosynthesis more efficient by absorbing the different wavelengths of the light. The single chlorophyll A molecule forms the reaction center. The reaction center is different in both the photosystem. In the photosystem 1, the reaction center chlorophyll A has an absorption peak at the 5 nanometer, hence it is called as the P phospho. While in the photosystem 2, it has an absorption maxima at the 6 nanometer, it is called as the 680 P 6. तो ये दिया हुआ है लाइट हार्वेस्टिंग कॉम्प्लेक्स दिया हुआ है, यहाँ पर दिया हुआ है फोटोन इस पर गिर रहा है, ये रिएक्शन सेंटर है और पिगमेंट्स मॉलिक्यूल्स हैं, और ये प्राइमरी रिसेप्टर जो कौन होता है, तो प्राइमरी रिसेप्टर जो होता है रिएक्शन सेंटर होता है। फिर आता है द इलेक्ट्रॉन ट्रांसपोर्ट के बारे में। तो इन द फोटोसिस्टम 2 द रिएक्शन सेंटर क्लोरोफिल absorbs the 6 nanometer wavelength of the red light, causing electron to become excited and jump into orbit farther from the atomic nucleus. These electrons are picked up by the electron effector, which passes them to the electron transport. फिर कहता है कि to an electron transport system consisting of the cytochromes. This movement of the electrons is downhill in terms of an oxidation reduction or the redox potential scale. The electrons are not used up as they pass through the electron transport chain but are passed on to the pigments of the photosystem PS1. Simultaneously, electrons in the reaction center of the PS1 are also excited when they receive the red light of the wavelength 4 nanometer and are transferred to another acceptor molecule that has a greater redox potential. These electrons then are moved downhill again, this time to the molecule of energy rich NADP+. The addition of these electrons reduces NADP+ to NADPH plus H+. This whole scheme of the transfer starting from the PS2 uphill to the receptor, down the electron transport chain to the PS1 excitation of the electrons, transfer of the another receptor, and finally downhill to the NADP+, reducing it to the NADPH plus H is called as a Z scheme due to its characteristic shape. This shape is formed when all the carriers are placed in a sequence on a redox potential scale पर। तो ये दिया हुआ है जेड स्कीम ऑफ द लाइट रिएक्शन। इधर ये यहाँ पर है LHC है, लाइट गिरा LH पर, तो इस पर H2O है जो यहाँ पर ब्रेक होता है, जो कि फोटोसिस्टम 1 है, तो 2 इलेक्ट्रॉन प्लस 2H प्लस ऑक्सीजन में टूट जाता है। फिर इलेक्ट्रॉन एक्सेप्टर फोटोसिस्टम 2 है ये, ADP प्लस इनऑर्गेनिक फॉस्फेट, इलेक्ट्रॉन ट्रांसपोर्ट फिर LH यानी कि दूसरे को देता है, लाइट होस्टिंग कॉम्प्लेक्स, ये फोटोसिस्टम 1 है। इलेक्ट्रॉन एक्सेप्टर यहाँ पर फिर FNR होता है जो NADP प्लस को NADPH में कन्वर्ट कर देता है। फिर आता है ये जेड स्कीम हो गया, क्योंकि जेड शेप है जो हमको ऐसे मिल रहा है। फिर आता है स्प्लिटिंग ऑफ द बाटर। कहता है कि यू वुड देन आस्क हाउ डाज फोटोसिस्टम 2 सप्लाई द इलेक्ट्रॉन्स कंटिन्यूसली, द इलेक्ट्रॉन्स दैट वेर मूव्ड फ्रॉम द फोटोसिस्टम 2 मस्ट बी रिप्लेस्ड, दिस इज अचीव्ड बाय द इलेक्ट्रॉन्स अवेलेबल due to the splitting of the barter. The splitting of the barter is associated with the photosystem 2. Barter is split into 2H plus O and the electron. This creates the oxygen, one of the net products of the photosynthesis. The electrons needed to replace those removed from the photosystem 1 are provided by the photosystem 2. तो इसमें बनता है 2H2O प्लस 4H प्लस प्लस O2 क्या बन जाता है, प्लस 4 इलेक्ट्रॉन बनता है। We need to emphasize here that the barter splitting complex is associated with the photosystem 2, which itself is physically located on the inner side of the membrane of the thylakoid. Then where are the protons and the O2 format likely to be released in the lumen or on the outer side of the membrane. फिर आता है साइक्लिक एंड नॉन साइक्लिक फोटोफॉस्फोरिलेशन। तो लिविंग ऑर्गेनिज्म हैव द कैपेबिलिटी ऑफ एक्सट्रैक्टिंग एनर्जी फ्रॉम द ऑक्सीडाइबल सब्सटेंसेस एंड स्टोर दिस इन द फॉर्म ऑफ द bond energy. A special substances like the ATP carry this energy in their chemical bonds. The process through which ATP is synthesized by the cells in the mitochondria and the chloroplast is named as the photophosphorylation. Photophosphorylation is the synthesis of the ATP from the ADP and inorganic phosphate in the presence of the light. When the two photosystems work in a series, first photosystem 2 and then the photosystem 1, a process called the non-cyclic photophosphorylation occurs. The two photosystems are connected through an electron transport chain as seen earlier in the Z scheme. Both the ATP and the NADPH plus H are synthesized by this kind of the electron flow. When only photosystem 1 is functional, the electron is circulated within the photosystem 1 and the phosphorylation occurs due to the cyclic flow of the electrons. A possible location where this could be happening as in the stroma lamella, where the membranes of the lamella of the grana have both photosystem 1 and the photosystem 2. The stroma lamella membrane lack the photosystem 2 as well as the NADP reductase enzyme. The excited electron does not pass on to the NADP plus but is cycled back to the photosystem 1 complex through the electron transport chain. The cyclic flow hence results only in the synthesis of the ATP but not of the NADPH plus H plus. Cyclic photosynthesis also occurs when the only light of the wavelength beyond 650 nanometer available for the excitation. तो ये दिया हुआ इधर साइक्लिक फोटोफॉस्फोरिलेशन दिया हुआ है, इधर ये लाइट हो गया और क्लोरोफिल P5 हो गया, इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम हो गया, इलेक्ट्रॉन एक्सेप्टर ये फोटो सिस्टम 1 का हो गया। फिर आता है केमियोस्मोटिक हाइपोथेसिस। तो let us now try and understand how actually ATP is synthesized in the chloroplast. The chemoosmotic hypothesis has been put forward to explain the mechanism like in the respiration in the photosynthesis 2. ATP synthesis is linked to the development of a proton gradient across the membrane. This time these are the membrane of the thylakoid. There is one difference though. Here the proton accumulation is towards the inner side of the membrane, that means in the lumen. In the respiration, proton accumulates in the intermembrane space of the mitochondria when the electrons move through the ETFs. Let us understand what causes the proton gradient across the membrane. We need to consider again the process that takes place during the activation of the electron and their transport to determine the steps that cause a proton gradient to develop. फिर आता है कि since the splitting of the barter molecule takes place on the inner side of the membrane, the proton or the hydrogen ions that are produced by the splitting of the barter accumulate within the lumen of the thylakoids में। दूसरा है कि as electrons move through the photosystem, protons are transported across the membrane. This happens because the primary receptor of the electron, which is located towards the outer side of the membrane, transfers its electron to an electron carrier but to an hydrogen carrier को, hence जो कि PQ होता है. Hence this molecule removes a proton from the stroma while transporting an electron. When this molecule passes on electron to the electron carrier on the inside of the membrane, the proton is released into the inner side of the lumen of the membrane. C process है कि the NADP reductase is located on the stroma side of the membrane along with the electrons that come from the receptor of the electrons of the photosystem 1. Protons are necessary for the reduction of the NADP plus to the NADPH plus H plus. These protons are also removed from the stroma. Hence, within the chloroplast, proton in the stroma decrease in the number, while in the lumen there is accumulation of the protons. This creates a proton gradient across the thylakoid membrane as well as a measurable decrease in the pH in the lumen. Why are we so interested in the proton gradient? This gradient is important because it is the breakdown of this gradient that leads to the synthesis of the ATP. The gradient is broken due to the movement of the proton across the membrane to the stroma through the transmembrane channel of the CF0 of the ATP synthase. The ATP synthase enzyme consists of the two parts. One called the CF0 is embedded in the thylakoid membrane and forms a transmembrane channel that carries out the facilitated diffusion of the proton across the membrane. The other portion is called the CF1 and protrudes on the outer surface of the thylakoid membrane on the side that faces the stroma. The breakdown of the gradient provides enough energy to cause a conformational change in the CF1 particle of the ATP synthase, which make the enzyme synthesis several molecules of the energy-packed ATP. Chemoosmosis requires a membrane proton pump, a proton gradient, and the ATP synthase. Energy is used to pump protons across a membrane to create a gradient or a high concentration of the protons within the thylakoid lumen. ATP synthase has a channel that allows the diffusion of the proton back across the membrane. This releases enough energy to activate the ATP synthase enzyme that catalyzes the formation of the ATP. फिर कहता है कि along with NADPH produced by the movement of the electrons, the ATP will be used immediately in the biosynthetic reaction taking place in the stroma responsible for the section CO2 the synthesis of the sugar. तो ये दिया हुआ है कीमोस्मोटिक हाइपोथेसिस में कैसे एटीपी बन रहा है, तो एटीपी सिंथेसिस थ्रू दी कीमोस्मोसिस। तो इधर है जो स्टोमा में लो H+ से लाइट गिरा, जैसे H2 है जो ब्रेक हुआ और इधर H+ कॉन्सेंट्रेशन बढ़ गया। उसी तरह से PQ है जो स्टोमा से H+ लेता है और लुमन में PQ H2 है जो लाके दे देता है, तो इधर भी उससे भी H+ बढ़ गया। इन दोनों से H+ बढ़ जाता है, जिससे लुमन में H+ ज़्यादा होता है, हाई H+ होता है। और इधर जो है H+ कम इससे भी हो रहा है कि FNR जो है NADPH को H+ को लेके NADP में H+ को जो दे रहा है स्टोमा में, जिससे NADPH बना रहा है, तो इससे है जो उधर भी घट रहा है H+। तो इससे कॉन्सेंट्रेशन ग्रेडियंट लुमेन में और स्टोमा के बीच है जो जनरेट हो गया, जिससे ये हो गया CF0 से जब H+ इधर जाएगा तो ATP सिंथेज यानी कि CF1 का है जो हेड घूम जाएगा, जिससे है जो ATP और इनऑर्गेनिक फॉस्फेट मिलके ATP बन जाएगा। फिर आता है वेयर आर द ATP एंड द NADPH जो बना है उसको यूज कहाँ करेंगे, तो वी लर्न दैट द प्रोडक्ट्स ऑफ द लाइट रिएक्शन आर द ATP, NADPH एंड O2. Of these, O2 diffuse out of the chloroplast, while the ATP and the NADPH are used to derive the process leading to the synthesis of the food, more accurately sugars. This is the biosynthetic phase of the photosynthesis. This process does not directly depend on the presence of the light, but is dependent on the products of the light reaction, that means the ATP and the NADPH, besides the CO2 and the H2O. You may wonder how this could be verified. It is simple. Immediately after the light becomes unavailable, the biosynthetic process continues for some time and then stops. If then light is made available, the synthesis starts again. फिर कहता है कि can we hence say that calling the biosynthetic phase as the dark reaction is a misnomer. फिर उसके बाद आता है कि let us now see how the ATP and the NADPH are used in the biosynthetic phase. Before earlier that CO2 is combined with the H2O to produce the CH2ON or the sugars. It was of the interest to the scientist to find out how this reaction proceeded, or rather what was the first product formed when the CO2 is taken into a reaction or reaction or fixed. Just after World War II, among the several efforts to put radioisotope to benzoes use, the work of the Melvin Calvin is exemplary. The use of the radioactive C14 by him in the algal photosynthesis study led to the discovery that this first CO2 fixation product was a 3-carbon organic acid. He also contributed to working out the complete biosynthetic pathway, hence it was called as the Calvin cycle after him. The first product identified by the 3-phosphoglyceric acid or in the short 3-PGA. फिर कहता है कि scientists also tried to know whether all plants have the PGA as the first product of the CO2 fixation, or whether any other product was formed in the other plants. Experiment conducted over a wide range of the plants led to the discovery of another group of the plants where the first of all product of the CO2 fixation was again an organic acid, but one had a 4-carbon among atoms in it. This acid was verified to be oxaloacetic acid or the OAA. Since then, the CO2 accumulation during the photosynthesis was said to be of the two main types. Those plants in which the first product of the CO2 fixation is a C3 acid, PGA, that means the C3 pathway, and those in which the first product by the C4 F8OA, that means the C4 pathway. These two groups of the plant foid other associated characteristics that we will discuss later. फिर कहता है the primary effector of the CO2 कौन सा होता है. So let us now ask ourselves a question that was asked by the scientists who were struggling to understand the dark reaction. How many carbon atoms would a molecule have which after fixing CO2 would have 3 carbons of the PGA? The study very unexpectedly showed that the acceptor molecule was a 5 carbon ketose sugar ribulose biphosphate RUBP. Did any of you think of this possibility? Don't worry, the scientists also took a long time and conducted many experiments to reach the conclusion. They also believed that since the first product was a C3 acid, the primary acceptor would be a 2 carbon compound. They spent many years trying to identify a 2 carbon compound before they discovered the 5 carbon RUBP. फिर कहता है the Calvin cycle. तो Calvin and his co-workers then worked out the whole pathway and showed that the pathway operated in a cyclic manner. The RUBP was regenerated. Let us now see how the Calvin pathway operated and where the sugar is synthesized. Let us at the outset understood very clearly that the Calvin pathway occurs in all photosynthetic plants. It does not matter whether they have the C3 or the C4 or any other pathway. For the ease of the understanding, the Calvin cycle can be described under the 3 stages: carboxylation, reduction and regeneration. तो पहला जो स्टेप आता है वो आता है कार्बोक्सिलेशन. Carboxylation is the fixation of the CO2 into a stable organic intermediate. Carboxylation is the most crucial step of the Calvin cycle, where the CO2 is utilized for the carboxylation of the RUBP. The reaction is catalyzed by the enzyme RUBP carboxylase, which results in the formation of the 2 molecules of the 3-PGA. Since the enzyme also has an oxygenation activity, it would be more correct to call it RUBP carboxylase oxidase or the Rubisco in the short. दूसरा स्टेप फिर आता है रिडक्शन. So these are a series of the reaction that lead to the formation of the glucose. The steps involve utilization of the 2 molecules of ATP for the phosphorylation and 2 of the NADPH for the reduction per CO2 molecule. Fixed. The fixation of the 6 molecule of the CO2 and the 6 turns of the cycle are required for the formation of the 1 molecule of the glucose from the pathway. फिर तीसरा है रीजनरेशन ऑफ द CO2 acceptor molecule RUBP is crucial if the cycle is to continue uninterrupted. The regeneration step also require 1 ATP for the phosphorylation to form the RUBP. फिर कहता है hence for every CO2 molecule entering the Calvin cycle, 3 molecules of the ATP and the 2 of the NADPH are required. It is probably to meet this difference in a number of the ATP and the NADPH used in the direct reaction that the cyclic phosphorylation take place. To make 1 molecule of the glucose, 6 turns of the cycles are required. Work out how many ATP and NADPH will be required to make 1 molecule of the glucose through the Calvin pathway. It might help you to understand all of this if you look at the what goes in the and what comes out at the Calvin cycle. तो 6 CO2 प्लस 18 ATP प्लस 12 NADPH मिल के एक ग्लूकोज बनाता है. ठीक है, तो एक ग्लूकोज को बनाने में 18 ATP और 12 NADPH लग रहा है कैल्विन साइकिल में. तो कैल्विन साइकिल दिखाया हुआ है. इसमें रुबलोज 1,5 बाइफॉस्फेट आ गया RUBP, इसमें से एक CO2 लगाएं तो कार्बोक्सिलेशन आया. कार्बोक्सिलेशन के बाद फिर 3 फॉस्फोग्लिसरेट 3 PGA बनाएं. उसके बाद 3 PGA जब बनाएं तो उसके बाद कार्बोक्सिलेशन करके 3 PGA बनाएं. उससे फिर उसके बाद फिर रिडक्शन किए. इसमें 2 मॉलिक्यूल ऑफ द ATP और 1 मॉलिक्यूल ऑफ द 2 मॉलिक्यूल ऑफ 3 इसमें फिर 2 मॉलिक्यूल ऑफ द ATP और 2 मॉलिक्यूल ऑफ द NADH लगा. फिर उसके बाद ट्राईफॉस्फेट करके रिएक्शन करके ट्राईफॉस्फेट बना जिसको हम PGAL पागल भी कहते हैं. और इससे फिर एक है जो ग्लूकोज, एक कार्बन नीचे चला गया जिससे फिर हम बाद में ग्लूकोज बना सकते हैं. और ग्लूकोज से फिर बाद में सुक्रोज बना के उसको ट्रांसपोर्ट कर सकते हैं फ्लोएम से, या स्टार्च बना के प्लांट को यूज भी करवा सकते हैं. तो डायरेक्ट लिख दिया है. उसके बाद फिर ट्राईफॉस्फेट को रीजेनेरेट करके भेज देंगे इधर जिसमें एक ATP खर्चा होगा और फिर हमको है जो RUBP है जो वापस मिल जाएगा. तो कैल्विन साइकिल के बारे में एनसीईआरटी क्या लिखता है? एनसीईआरटी कैल्विन साइकिल के इसके बारे में लिखता है कि द कैल्विन साइकिल proceeds in the 3 stages: carboxylation during which the CO2 combines with the RUBP, reaction during which the carbohydrate is formed at the expense of the photochemically made ATP and the NADPH, तीसरा है regeneration during which the CO2 acceptor ribulose 1,5 is formed again so that the cycle है जो कंटिन्यू हो सके. इसके बाद फिर आता है C4 pathway. तो plants that are adapted to the dry tropical regions have the C4 pathway mentioned earlier. Though these plants have the C4-oxaloacetic as the first CO2 fixation product they use, the C3 pathway or the Calvin cycle as the main biosynthetic pathway, then in what way are they different from the C3 plants? This is a question that you may reasonably ask. C4 plants are special. They have a special type of the leaf anatomy. They tolerate higher temperature, therefore response to high light intensities. They lack a process called the photosynthesis and have greater productivity of the biomass. Let us understand each one by one. फिर कहता है कि study the vertical section of the leaves, the one of a C3 plant and the other of a C4 plant. Do you notice the difference? Do both have the same type of the mesophiles? Do they have the similar cells around the vascular bundle sheath? The particularly large cells around the vascular bundles of the C4 plants are called like the bundle sheath cells, and the leaves which have the such anatomy are called like the cranch anatomy. Cranch means the wreath and is a reflection of the anatomy arrangement of the cells. The bundle sheath cells may form the several layers around the vascular bundles. They are characterized by having a large number of the chloroplasts, thick wall, impervious to the gas exchange, and no intercellular species. You may like to cut a section of the leaves of the C4 plant, maize or the sorghum to observe the cranch anatomy and the distribution of the mesophile cells. It would be interesting for you to collect leaves of the diverse species of the plant around you and cut the vertical section of the leaves. Observe under the microscope, look for the bundle sheath around the vascular bundle. The presence of the bundle sheath would help to identify the C4 plants. फिर कहता है कि now study the pathway shown in the figure 13.9. This pathway that has been named as the hatch flag pathway is again a cyclic process. Let us study the pathway by listing the steps. The primary CO2 substrate is a 3-carbon molecule phosphoenol pyruvate PEP and is present in the mesophile cells. The enzyme responsible for this fixation is a PP carboxylase or the pepcase. It is important to register that if mesophile cells lack the visco enzyme, the C4 acid OA is also formed in the mesophile cells. It then forms another 4-carbon compounds like the malic acid or the aspartic acid भी कहता है NCERT in the mesophile cells itself, which are transported to the bundle sheath cell. In the bundle sheath cell, these C4 acids are broken down to release the CO2 and the 3-carbon molecule. फिर कहता है the 3-carbon molecule is transported back to the mesophile cell where it is converted to the PP again, thus completing this cycle. The CO2 released in the bundle sheath cell enters the C3 or the Calvin's pathway, a pathway common to the all plants. The bundle sheath cells are rich in an enzyme called the ribulose biphosphate carboxylase oxidase ribosco but lack the pepcase. Thus, the basic pathway that results in the formation of the sugar, the Calvin pathway, is common to the C3 and the C4. Did you note that the Calvin pathway occurs in all the mesophile cells of the C3 plant? In the C4 plant, it does not take place in the mesophile cells but does so in the bundle sheath cells में. फिर आता है ये डायग्रामेटिक व्यू दिए हुए है, इधर डायग्रामेटिक रिप्रेजेंटेशन ऑफ दी हैच फ्लैग पाथवे या C4 साइकिल का दिए हुए है, जिसमें यहाँ पर मेसोफाइल सेल्स हो गया, ये बंडल स्विच सेल्स हो गया. तो एटमॉस्फेरिक CO2 है जो मेसोफाइल सेल्स में आया, जहाँ वाटर के साथ मिलकर बाइकार्बोनेट बना, बाइकार्बोनेट और फॉस्फोनिल पायरुवेट PP को मिलाकर फिक्सेशन किए, मैलिक एसिड बनाया, मैलिक एसिड का ट्रांसपोर्ट किए और फिर वहाँ C4 एसिड मैलिक एसिड को OA में कन्वर्ट कर दिए. OA से एक है जो CO2 गया, डीकार्बोक्सिलेशन किए और उसको है जो कैल्विन साइकिल में भेज दिए, जहाँ उसको है जो यूज करेगा फिर डायरेक्ट रिएक्शन में. फिर है जो OA है जो से जब एक CO2 निकाल रहे थे तो OA खुद है जो C3 एसिड पायरुविक एसिड में कन्वर्ट हो गया. पायरुविक एसिड को हम ट्रांसपोर्ट करके फिर से मेसोफाइल सेल्स में भेज दिए. मेसोफाइल सेल्स में जो है उसको है जो रीजेनरेट किए, 2 ATP खर्चा करके उसको फॉस्फोइनोल पायरुवेट में फिर से है जो बना दिए और ये साइकिल इस तरह से फिर से है जो चलते रहेगा. फिर आता है फोटो रेस्पिरेशन. तो NCERT कहता है कि लेट अस ट्राई एंड अंडरस्टैंड वन मोर प्रोसेस दैट क्रिएट्स एन इंपॉर्टेंट डिफरेंस बिटवीन द C3 एंड द C4 प्लांट्स, फोटो रेस्पिरेशन. टू अंडरस्टैंड द फोटो रेस्पिरेशन वी हैव टू नो a little more about the first step of the Calvin 5 pathway, the first CO2 fixation step. This is the reaction where the RUP combines with the CO2 to form the 2 molecule or the 3 PGA that is colored by the ribosco. तो इसमें क्या आ रहा है कि RUP प्लस CO2 रिबोस्को के साथ मिलकर 2 मॉलिक्यूल है जो PGA का बना रहा है और जो PGA है ये 3 कार्बन का मॉलिक्यूल होता है. तो रिबोस्को दैट इज द मोस्ट डोमिनेंट एंजाइम इन द वर्ल्ड. डू वंडर व्हाई इज कैरेक्टराइज्ड बाय द फैक्ट दैट इज एक्टिव साइट कंबाइंड टू द बोथ CO2 एंड द O2, यानी कि पुजारी भी है और बेबड़ा भी है. Hence the name. Can you think how this could be possible? Ribosco has a much greater affinity for the CO2 when the CO2 O2 is nearly equal. Imagine what would happen if this were not so. This binding is competitive. It is the relative concentration of the O2 and the CO2 that determines which of the two will bind to the enzyme. फिर आता है In the C3 plant, some O2 did not bind to the RUBSCO, hence the CO2 fixation is decreased. Here the RUEP, instead of being converted to 2 molecules of the PGA, bind with the O2 to form 1 molecule of the phosphoglycerate and the phosphoglycolate 2 carbon in a pathway called the photorespiration. In the photorespiratory pathway, there is neither synthesis of the sugar nor of the ATP. Rather, it results in the release of the CO2 with the utilization of the ATP. In the photorespiratory pathway, there is no synthesis of the ATP or the NADPH. The biological function of the photorespiration is not known yet. फिर कहता है कि इन द C4 plant photorespiration does not occur. This is because they have a mechanism that increase the concentration of the CO2 at the enzyme site. This reaction takes place when the C4 acid from the mesophile is broken down in the bundle switch cell to release the CO2. This results in the increasing the intercellular concentration of the CO2. In turn, this ensures that the RUBSCO function as a carboxylase, minimizing the oxygenase activity. Note that you know that the C4 plant lacks the photorespiration. You probably can understand why productivity and yields are better in these plants. In the addition, these plants show the tolerance to the higher temperature. Based on the above discussion, can you compare plants showing the C3 and the C4 pathway? Use the table format given and fill in the information. तो इस टेबल को फिल करेंगे तो इसमें पूछ रहा है कि सेल बीच टाइप सेल जो टाइप हो गया कि सेल टाइप इन बीच तक कैल्विन साइकिल टेक्स प्लेस। तो इसमें होता है मेसोफाइल सेल्स में और इसमें होता है कैल्विन साइकिल किसमें होता है। तो इसमें कैल्विन साइकिल जो होता है बंडल सेल्स में होता है। सेल इन बीच द इनिशियल कार्बोक्सिलेशन टेक प्लेस। तो इसमें जो होता है इसमें इनिशियल कार्बोक्सिलेशन मेसोफाइल सेल्स में होता है। इसमें इनिशियल कार्बोक्सिलेशन मेसोफाइल इसमें भी होता है। हाउ मनी सेल डाई द लीफ हैव दैट फिक्स द CO2। तो इसमें 1 और इसमें 2 हो जाएगा। फिर आता है विच इज द प्राइमरी CO2 रिसेप्टर। तो इसमें जो है RUEP है, इसमें है जो PP है। फिर नेम ऑफ द कार्बन इन द प्राइमरी CO2 रिसेप्टर। तो इसमें है जो 5 है और इसमें है जो 3 है। PP में 3 कार्बन होता है। फिर व्हाट इज द प्राइमरी CO2 व्हाट इज द प्राइमरी CO2 फिक्सेशन प्रोडक्ट। इसमें बनता है PGA और इसमें बनता है OAA। फिर नंबर ऑफ द कार्बन इन द प्राइमरी CO2 फिक्सेशन प्रोडक्ट। इसमें 3 होता है, इसमें है जो 4 होते हैं। फिर डाई द प्लांट हैव द RUBSCO। तो इसमें F, इसमें भी F, इसमें बंडल सेल में हो जाएंगे C4 में। डाई द प्लांट हैव द पेप्टाइड। तो इसमें नो, लेकिन इसमें F हो जाएगा। विच सेल इन द प्लांट हैव द RUBSCO। तो इसमें मेसोफाइल सेल्स में और इसमें है जो बंडल सेल्स में। CO2 फिक्सेशन रेट अंडर हाई लाइट कंडीशन में। तो CO2 फिक्सेशन रेट अंडर हाई लाइट कंडीशन में क्या हो जाएगा। तो अंडर हाई लाइट कंडीशन में है जो इसमें भी हाई होगा और इसमें भी हाई होगा। फिर विदर फोटो रिस्पिरेशन इज प्रेजेंट एट द लो लाइट इंटेंसिटी में। तो लो लाइट इंटेंसिटी में फोटो रिस्पिरेशन नहीं होगा और इसमें तो फोटो रिस्पिरेशन होता ही नहीं है। विदर फोटो रिस्पिरेशन इज प्रेजेंट एट हाई लाइट इंटेंसिटी। इसमें यस, लेकिन इसमें नहीं होगा। विदर फोटो रिस्पिरेशन वुड बी प्रेजेंट एट लो CO2 कंसंट्रेशन में। तो इसमें है जो फोटो रिस्पिरेशन हो जाएगा, लेकिन इसमें है जो नहीं होगा। विदर फोटो रिस्पिरेशन वुड प्रेजेंट एट हाई CO2 कंसंट्रेशन में। तो हाई CO2 कंसंट्रेशन में नहीं होगा, लेकिन और इसमें तो फोटो रिस्पिरेशन होता नहीं है। तो टेम्परेचर ऑप्टिमम इसे 20 से 25 चाहिए और इसे 30 से 40 चाहिए। इसका एग्जाम्पल में आ जाता है मेज सोरगम आ जाता है और सुअरकेन आता है और इसके एग्जाम्पल में भीट वगैरह आ जाते हैं। फिर कहता है फैक्टर्स अफेक्टिंग द फोटोसिंथेसिस। तो एनसीईआरटी कहता है an understanding of the factors that affect the photosynthesis is necessary. The rate of the photosynthesis is very important in determining the yield of the plant, including the crop plant. Photosynthesis is under the influence of the several factors, both internal and external. The plant factors include the number, size, age, and the orientation of the leaves, mesophyll cells, and the chloroplast, and the internal CO2 concentration and the amount of the chlorophyll. The plant or the internal factors are dependent on the genetic predisposition and the growth of the plant. The external factors would include the ability of the sunlight, temperature, CO2 concentration, and the water. And as a plant photosynthesizes, all the factors will simultaneously affect its rate. Hence, those several factors interact and simultaneously affect the photosynthesis or the CO2 fixation. Usually, one factor is the major cause or that one that limits the rate. Hence, at any point, the rate will be determined by the factors available at the suboptimal levels. फिर एनसीईआरटी कहता है कि when several factors affect any biochemical process, ब्लैकमन्स लॉ 1905 में लॉ ऑफ लिमिटिंग फैक्टर्स दिया था come into the fact. This states that the following: if a chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearest to the minimum value. It is the factor which directly affects the process if its quantity is unchanged. जैसे फॉर एग्जाम्पल कहता है कि डिस्पाइट द प्रेजेंस ऑफ द ग्रीन लीफ एंड ऑप्टिमल लाइट एंड CO2 कंडीशन द प्लांट में नॉट बी photosynthesize if the temperature is very low. This leaf if given the optimum temperature will start photosynthesizing. फिर आता है लाइट, तो कहता है we need to distinguish between the light quality, light intensity and the duration of the exposure to the light by discussing the light as a factor that affects the photosynthesis. There is a linear relationship between the incident light and the CO2 fixation rate at a low light intensity. At the higher light intensity, gradually the rate does not show further increase as the other factors become limiting. But it is interesting to note that the light saturation occurs for the 10% of the full sunlight, hence except for the plant in the shade or in the dense forest, light is really limiting for the factor in the nature. Increase in the इंसिडेंट लाइट बियॉन्ड द पॉइंट कॉज द ब्रेक डाउन ऑफ द क्लोरोफिल एंड डिक्रीज इन द फोटोसिंथेसिस। तो ये दिया हुआ है ग्राफ ऑफ द लाइट इंटेंसिटी ऑन द रेट ऑफ द फोटोसिंथेसिस पर दिया हुआ है। ये लाइट इंटेंसिटी हो गया, ये रेट ऑफ द फोटोसिंथेसिस हो गया। यहाँ पर थोड़ा सा इधर से स्टार्ट करना चाहिए था। यहाँ पर हो गया सैचुरेशन पॉइंट, यहाँ पर इसको कहेंगे कंपनसेशन पॉइंट। फिर आता है कार्बन डाइऑक्साइड कंसंट्रेशन, तो इसके बारे में बोलते हैं एनसीईआरटी कार्बन डाइऑक्साइड इज द मेजर लिमिटिंग फैक्टर फॉर द फोटोसिंथेसिस। द कंसंट्रेशन ऑफ द CO2 is very low in the atmosphere between 0.03 and the 0.04%. The increase in the concentration up to the 0.05% can cause an increase in the CO2 fixation rate. Beyond this level can become damaging over the longer periods. The C3 and the C4 plant responds differently to the concentration. At low light concentration, neither group respond to the high CO2 condition. At the high light intensity, both C3 and the C4 plants show the increase in the rate of the photosynthesis. But important to note is that C4 plants show the saturation at about 240 microliter per liter, where the C3 plants show the increased CO2 concentration and the saturation is seen only beyond 4 microliter per liter. This the current ability of the CO2 level is limiting to the C3 plant. फिर आता है द फैक्ट दैट द C3 plant respond to the higher CO2 concentration by showing the increased rate of the photosynthesis leading to the higher productivity has been used for the some greenhouse crops such as the tomatoes and the bell pepper. They are allowed to grow in the carbon dioxide rich atmosphere that leads to the higher yields. फिर आता है टेम्परेचर, तो the dark reaction being enzymatic are temperature controlled, though the light reactions are also temperature sensitive, they are affected by the much lesser extent. The C4 plant respond to the higher temperature and for higher rate of the photosynthesis, while the C3 plant have a much lower temperature optimum. The temperature optimum for the photosynthesis of the different plant also depends on the habitat that they are adapted to. Tropical plants have a higher temperature optimum than the plants adapted to the temperate climates. फिर आता है वाटर, तो कहता है कि even though butter is one of the reactants in the light reaction, the effect of the butter as a factor is more though its effects on the plant rather than directly on the photosynthesis. Butter stress causes the stomata to close, hence reducing the CO2 availability. Besides, butter stress also make the leaves wilt, thus reducing the surface area of the leaves and their metabolic activity as well. फिर इसका समरी है कि green plant make their own food by the photosynthesis. During this process, carbon dioxide from the atmosphere is taken in the leaves through the stomata and used for the making of the photocarbohydrates, principally glucose and the starch. Photosynthesis takes place only in the green parts of the plant, mainly the leaves. Within the leaves, the mesophyll cells have a large number of the chloroplasts that are responsible for the CO2 fixation. Within the chloroplast, the membranes are set for the light reaction, while the chemosynthetic pathway occurs in the stroma. Photosynthesis has two stages, the light reaction and the carbon fixing of reaction. In the light reaction, the light energy is absorbed by the pigments present in the antenna and the funneled to the special chlorophyll, a molecule called the reaction center chlorophylls. There are two photosystems, PS1 and the PS2. PS1 has a phosphor nanometer absorbing chlorophyll A, p-phosphor molecule at its reaction center, while the PS2 has a p-Chesophy reaction center that absorbs the red light at the Chesophy nanometer. After absorbing light, electrons are excited and transported through the PS2 and the PS1 and finally to the NAD, forming NADH. During this process, a proton gradient is created across the membrane of the thylakoid. The breakdown of the proton gradient due to the movement through the F0 part of the ATPase enzyme release the enough energy for the synthesis of the ATP. A splitting of the butter molecule is associated with the PS2, resulting in the release of the O2 protons and the transfer of the electrons to the PS2. In the carbon fixation cycle, CO2 is added to the enzyme और उसको to a 5 carbon compound RUP that is converted to molecule of the 3 carbon PGA. This is then converted to sugar by the Calvin cycle and the RUP is regenerated. During this process, ATP and the NADP is synthesized in the light reaction are utilized. RUBSCO also catalyzes a beautiful oxidation reaction in the C3 plants called the photorespiration. Some tropical plants show a special type of the photosynthesis called the C4 pathway. In these plants, the first product of the photosynthesis takes place in the mesophile is a 4-carbon compound. In the biosphere, the calcium pathway is carried out for the synthesis of the carbohydrate.